drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I'm going to take just a minute to tell you about Spores MD, your trusted source for premium mushroom products designed for research and educational purposes. At sporesmd.com, you can discover pharmaceutical-grade mushroom liquid cultures featuring dozens of unique strains of psilocybe mushroom species ideal for microscopy studies and mycology enthusiasts. They also offer culinary liquid cultures for gourmet mushroom enthusiasts, plus innovative options like nootropics gummies and Amanita muscaria tinctures, all produced in ISO 7 certified clean rooms with rigorous testing for viability, sterility, and purity. With a 100% satisfaction guarantee, Spores MD ensures high-quality products free from contaminants and without traces of controlled substances. Whether you're interested in scientific exploration or simply curious about the fascinating world of fungi, Spores MD provides reliable tools to support your interests, always in compliance with applicable laws. Head to sporesmd.com today and use the discount code TRICHOMES, TRIKOMES, for 20% off your purchase today. So go to sporesmd.com and check out what they have. Danielle Stolzenberg, thank you for joining me.
Thank you so much for having me.
Do you want to start off by just telling everyone a little bit about who you are and what your lab studies?
Sure. Yeah, I'm an associate professor at the University of California, Davis, and my lab is interested in the neurobiology of maternal care broadly. We've been really focused on understanding how experiences with infants in the postpartum period can lead to long-term changes in brain and behavior, and more recently, how maybe experiences in the postpartum period more generally can impact long-term behavior and plasticity.
So when we think about maternal care behavior, what is maternal care behavior as a neuroscientist like yourself would define it? We all probably have an intuitive idea of what this is. Obviously, mothers tend to care for their young in some way, shape, or form across different species. That manifests differently in different species. But when we think about it in mammals, in particular, what is it in mammals and how does it maybe differ on average from other types of creatures?
Great question. As you mentioned, we think about it as really ensuring the survival of offspring. And in mammals, maternal care is a defining characteristic, actually, of mammalian behavior. So there's quite a bit of conservation across species, but there's a few categories of behavior that moms tend to show. So they provide food in the form of milk and lactation, also a defining characteristic of mammals. They will protect their infants if there's any sort of threat from a predator, even a conspecific. They'll also provide warmth, so they'll build a nest. This is particularly important for rodents as they tend to be they tend to have not a lot of capability to regulate their own body temperature when they're first born. So the nest is really important for that. So they'll keep them warm. They'll also provide grooming behavior, which in rodents functions to not only impact emotional development, but also um, physically to help them urinate and defecate actually in the first few days of life. So
Hmm.
these behaviors are absolutely critical. Pups are born, again, for rodents, pups are born what we call altricial, incredibly immature. Their eyes are closed. They can smell, but they have limited sensory and motor capabilities. And if they were to get lost from the nest, so let's say they're nursing and mom goes to forage a bit, they can kind of fall off and they wouldn't be able to find their way back to the nest. So moms also retrieve pups where they'll pick the pup up in the mouth and bring it back to the nest to make sure that they're safe. So those are, I sort of described it in the context of rodents, but many of these behaviors are conserved across species. So um, non-human primates, humans, um, also nurse, also carry infants around, also protect the young. And so depending upon the sort of development of the species, development of the young at birth in a given species, you can see variations in care. But overall, the care is really what infants will need in order to survive, particularly in the very early postpartum period.
Mhm. And so when I think about sort of the full spectrum of the animal kingdom, there's obviously species that don't do any parental care whatsoever. You know, a salmon will lay the eggs. There's no parental input whatsoever. I can think of non-mammalian species, though, that have a lot of pretty elaborate parental care. Crocodiles and alligators,
Mhm.
one or both of them, right? The little babies will hide in the mom's mouth. So they will protect them. They will guard the nest. They will help the thermal regulate, keep the eggs the appropriate temperature, those types of things. So it's not that maternal care behaviors don't exist outside of mammals, and obviously birds, they guard the nest, they lay on the eggs, they feed the babies. But I guess one of the things that probably defines mammals is maybe implied by the name. They mammalian young need the mother there for all of those other reasons, but also because they have to consume milk. Got it. So maybe there's just sort of a level of intimacy and a level of care that tends to be higher in mammals on average than other creatures.
That's a great question. I don't know if I would phrase it like that actually, because so I think the way I think about it, caregiving behaviors are highly conserved
Mm-hmm.
across
Mm-hmm.
many species, as you mentioned, outside of mammals. And actually, when we think of maternal behavior specifically, I tend to think about mammals because about 95% of mammalian species, mom is the sole caregiver. And I think that's very much related to the fact that mammals lactate, and that becomes an important part of caregiving. But in non-mammalian species, oftentimes the dad is can be the primary
Hmm.
caregiver. And so there's a whole slew of behaviors across many species that animals can um, conduct in order to make sure that the offspring survive anywhere from the time of, let's say, laying eggs through the early period after they're hatched. And so it just sort of depends on the species what kinds of caregiving behaviors occur.
Hmm. And throughout this discussion, we'll primarily talk about rodents and the things that you guys study in the lab, but we'll probably also tie it to behaviors in other animals and, of course, human beings themselves. We have maternal care behavior and parental behavior. So we'll tie to those things, I think, and weave that together as we go throughout this. And people will obviously be thinking about the human parallel to a lot of the stuff we discuss in rodents. And, we'll add some color there as needed. But starting out with rodents, because that's what's studied in the lab by people like you, what are can you just kind of give us a survey of when you have a new mother, a mouse, what are the core maternal care behaviors she tends to exhibit? What are the basic things she does?
So shortly after, so first of all, moms are actively engaged in caregiving behavior from the moment the pups are being birthed. So they will eat the placenta, start grooming the pups immediately. And then actually, this will happen a couple of days even before mom gives birth. She'll begin to build a nest, gathering nest materials so that she can keep the pups warm right after birth. She will, as I mentioned before, retrieve pups that should become displaced from the nest. This is actually my favorite behavior and the behavior that I've studied the most in my lab, where there it could be a number of reasons why pups become displaced. Pups could be suckling, and if mom gets up quickly, sometimes they're sort of still hanging on and then they might fall off. As they get a little bit older and mobile, they can kind of start to leave the nest. Regardless of what happens, mom will pick them up and carry them back to the nest to make sure that they're safe. And rats are really good at this behavior. Actually, we sort of joke in the lab sometimes that seems like they can count, because if you, as the experimenter, separate pups from the nest and sort of scatter them throughout the cage to sort of stimulate this pup retrieval behavior, you'll see this very stereotypic response. Mom will come out, she'll pick each pup up, bring it back to the nest, and then when she's done, she'll sort of circle around the cage, making sure that she's got everyone before she eventually settles back in the nest. And then in the nest, moms display nursing behavior, as we mentioned, in order to keep the pups fed. There's a number of different postures that they can take over the pups when they're engaged in nursing behavior. Nursing behavior is largely reflexive. So one of the things that we tend to look for in my lab, because we study stress during the postpartum period, is we really look to see if mom's even giving pups a chance to get near the nipples, because once mom's kind of over the pups, it's actually stimulation from the infants that causes her to go into different nursing postures. And mom really responds to the infant needs quite a bit as well. So one of the things we've learned in the last decade or so is that pups emit ultrasonic vocalizations that we're not able to audibly hear. But mom can hear, and they emit different types of vocalizations depending upon what they might need at any given time. So some vocalizations are distress calls. If they get separated, that's one of the ways that mom can find them. But even in the nest, they'll emit different vocalizations that will cause mom to shift from nursing to grooming and so forth. And then, as I mentioned, they also groom the pups. So grooming behavior, early on, it functions to help the pups urinate and defecate. And moms display this behavior quite a bit. So they lick the body, they lick the anogenital region, and as pups get older, they continue to exhibit this sort of body grooming behavior. And there's been a lot of work done looking at the relationship between that sort of grooming, physical contact, and various outcomes in pups once they're adults.
So at one level, it's sort of all the things that you would expect. It's a lot of similar things to what humans do. The mother's providing warmth. She's providing security. She will get everyone back together if they're displaced or separated. She will provide food, obviously, and grooming and things like that. So a lot of familiar behaviors there that we see in a lot of mammals. And it sounds like there's a lot of specific and clear sensory feedback from infant to mother and vice versa that sequences and structures the behavior appropriately. She can hear them. It's kind of like an infant. You mentioned they have these different vocalizations. Infants cry in different ways if they're hungry versus uncomfortable and things like this. So there's a lot of communication here happening.
Absolutely. And moms learn over time how to respond as pups need. This is not my area of expertise or my work, but there's been a lot of really beautiful work that's been done by Robert Liu and his lab, looking at that this sort of interaction between pups calls and mom's responses, and plasticity in the auditory cortex in both postpartum females and virgin females, or females that have never had sexual experience but are capable of caring for infants.
Yeah, and that's an interesting thing. So like a virgin female has never had pups of her own, but they will often exhibit maternal behavior. This brings us to the concept of, how much of this is instinct and how much of it is learned. Presumably there's some amount of both, but a lot of this is probably pretty instinctive, because when the mothers have their first litter, they do all these things. The mice aren't going to, maternal care classes or, calling up their grandma and asking her how to do things. So can you start to talk about that a little bit? How much of this maternal care behavior is instinctive? How much of it is learned? And how do we start to study that in the lab?
Yeah, that's a great question. And I think about this a lot and have done work to address this question really over the course of my career. But, the way I like to describe it is I think about the capacity for care as innate, but the caregiving behaviors themselves have to be turned on. And so I imagine that, the sort of circuitry is there. We know that this from research that the circuitry is there. The circuitry is highly conserved in males and in females, and across species. And yet, whether or not caregiving behavior will occur depends oftentimes on hormone context, social experience, variability across species. Some species are relatively responsive to infants, regardless of their life experiences or whether they've had exposure to pregnancy hormones. Other times, certain species are really reliant on hormonal changes that happen really right around the time of birth to synchronize the onset of care. And then, even though these behaviors are turned on at the time of birth, and it really does sort of look like this magic switch, there's still learning that's occurring.
Right.
And actually, I think about those hormonal changes around the time of birth as really being this sort of really allowing the experience of interacting with infants to produce these long-term changes. So it's
Right.
it's a facilitatory effect. It's not an automatic effect. So, for example, if in rats, where you really need hormonal changes in order to get a relatively quick display of caregiving behavior, if you allow females to experience pregnancy and birth, but you immediately separate them from their infants, and then you give infants back to see if they'll display caregiving behaviors, they look just like a virgin female. So it's not the hormonal changes alone
Right.
that are producing caregiving behavior. It's sort of the combination of these hormonal changes, the experience of interacting with pups, and that really seems to lead to this consolidation.
Right.
Without those hormonal changes, you can sometimes also get experience-induced effects. So this is something I've been really interested in my lab. Oftentimes, you just need more experience with
Right.
infants before you get those same changes. So it sort of expedites, the
Right.
word I was looking for, really
Yeah.
expedites the experience-dependent effects.
Yeah. So we can, roughly speaking, we can think of hormonal changes that are presumably happening from the moment of the sexual act through the pregnancy itself. It's re-sculpting circuits in ways that enable, it makes the female more sensitive to certain environmental stimuli, like the vocalizations from the pups, the sensations of the pups, and all of those things. It's not necessarily turning on all of the maternal care behaviors wholesale. It's just making her more sensitive to inputs or increasing or decreasing certain behavioral tendencies she might have that are then further refined by experience. So the female's probably made more interested in things like the smell, the sight, and the feeling of the pups than she would have otherwise been. But then as she actually has experience with actual pups after they're born, she does get better at doing those things. You can, if we anthropomorphize for a minute, you can imagine a new mother being very interested in these ultrasonic vocalizations, but maybe not quite knowing what to do. But then she does it the first time, and she gets a reinforcement signal that says, "Ah, yes, that feels good. That was what was supposed to happen." So the sensitivity to certain stimuli, the tendency to engage in certain behaviors is what's being switched on, if you will, by hormonal signals. But that's not sufficient on its own. You need experience to actually refine the behavior.
Yes, that's exactly right. I love the way that you describe that. It's a great description. There are all kinds of changes that are happening across pregnancy. And really, when we think about the hormonal changes that are expediting these processes, they're happening really at the very end of pregnancy, right before birth. But one of the really cool series of studies that has been conducted in rats. So rats, as I mentioned, are not particularly interested in infants. They actually find the smell of infants to be aversive. And so sort of like, if a virgin female is sniffing a pup, there's a very
I see.
easy
They don't
to
like it. They don't
observe
want to
sort
go
of,
by it. Yeah.
no, they're really not interested. And actually they show this work was done by Alison Fleming. They show active avoidance. So if you put a pup next to them, they try to get as far away as they possibly can in the cage. And one of the things that Alison Fleming in her lab showed in a series of really beautiful papers is that this is mediated by the smell, by the olfactory inputs, and actually the valence of those olfactory inputs changes across pregnancy. So when females get closer to the time of birth, not only do they no longer find those smells aversive, but now they
Wow.
actually find those smells to be attractive
Yeah.
and they want to
Yeah.
interact.
It's
Yeah,
yeah, it's hard not to draw the human parallels there. We can
right.
probably all think of examples where a baby might produce odors that we all find aversive, but the mother gets very interested in those smells in a different way.
That's exactly right.
So there's also parallels here that we will come back to in more detail later with the concept of sensitive periods in development. So oftentimes when we talk about critical or sensitive periods in development, we're talking about childhood or the early phases of juvenile development in an animal. When you're younger, you're a little bit more plastic in different ways. You learn faster. Your brain's squishier, if you will. Critical periods for language acquisition, sensitive periods for different behaviors. The juvenile brain is different than the adult brain, but pregnancy itself and the time of development around that for the mother can also be thought of as a sensitive period. I think the mother's body has to become re-sculpted and rewired, all over the place to accommodate the developing fetus, to change her behavior so that she can do these maternal care behaviors and take care of the young after they're born. And so pregnancy itself is very much like a sensitive period. And, when we look under the hood a little bit, there's a lot of hormonal changes that are enabling that are changing in very interesting and dynamic ways, not unlike the hormonal changes that, might instigate something like puberty or other sensitive periods of life. So I want to talk a little bit more about the nuts and bolts here. So after a female rodent gets pregnant and she goes through the pregnancy and then she has the pups and she starts doing these maternal care behaviors, can you give us a sense for some of the major hormonal changes that underlie that? And then from there, we'll probably go into some of the circuitry that those hormones are acting on to actually, flip the switch, so to speak, on some of these behaviors.
Yeah, absolutely. So that's probably the two biggest hormones that we think about when we think about hormonal changes during pregnancy, um, are estradiol, which would be part of what we would think of maybe more commonly as estrogen. So really, there is no such thing as estrogen. It's estrogens as a class of hormones:
Mhm.
estradiol, estrione, and estriol. But estradiol is really the primary one. Um, and then progesterone is also changing. And progesterone, the hormone progesterone, literally means progestation. So progesterone is climbing throughout pregnancy, as is estradiol. And then around the time of birth, there's just this fallout. Everything just comes sort of crashing down. And actually, the drop in progesterone is really necessary in order for birth to take place, because progesterone is sort of one mechanism, peripherally speaking, is it's sort of in antagonizing oxytocin and preventing an early expulsion of the fetus. And
Hmm.
so you need progesterone to sort of drop out if birth is going to happen.
Got it. You need it to go
Um,
high and then drop off.
and come back down. Exactly. And estradiol is still um, higher than progesterone at that point, but both are really kind of going to be dropping out quite a bit in the around the time of birth, and shortly thereafter. So these hormonal changes, I really can't emphasize how large they are. We sometimes call them supraphysiological because, we tend to think about like what, in science, what is in the physiological range, like what would be natural in a bodily system. And oftentimes when we talk about pregnancy hormones, we call them supraphysiological because they're changing on a hundredfold
Yeah. Yeah.
scale.
Obviously it's perfectly natural, it's perfectly physiological, but
Exactly.
it's such a bigger change compared to any other aspect of life that an animal would be in that we call it, a super stimulus.
Exactly. Exactly. Yeah.
Okay, so when people hear the word estrogen, this is really a class of compounds, but when you hear estrogen did this, typically people what they really mean is estradiol. That's sort of the major estrogen that gets talked about. There are other ones obviously. So you got estradiol, you've got progesterone. They are changing throughout the pregnancy, but roughly speaking, they're going way up and then they're coming back down later at the time of birth. The absolute levels and the relative levels of these things matter. And then estradiol and progesterone, these are the things that wax and wane, not maybe as much, but throughout the menstrual cycle. And they're what we
Mhm.
find in hormonal birth control as well, right? So these are the
Correct.
pregnancy hormones, let's say.
Hmm. Yes, they fluctuate at many times throughout life, and certainly in a cyclical way throughout
Mhm.
menstruation.
So as the pregnancy is progressing, you've got increases in hormones like estradiol and like progesterone. And obviously those things are going to regulate, the birth, the placenta, like all of the stuff happening down there. But when we think about what's happening up in the brain of the mother, how are these hormones actually doing brain stuff? How are they getting into the brain? And where in the brain are some of the key sites of interaction where you've got plasticity happening and you've got a rewiring of the mother's brain that's preparing her for her maternal care behavior that's coming up?
Yeah, that's a great question. So they're primarily, estradiol and progesterone are primarily produced in the ovary, but they are what we call steroid hormones, which means they're fat soluble. They travel throughout the blood. They easily cross the blood-brain barrier. And really, they can be orchestrating changes throughout the brain and the body. But the brain is very responsive to these two hormones because there are receptors for these hormones that exist throughout all of the regions of the brain that people really across neuroscience, across fields in neuroscience like to study. And so these receptors can exert what we would call slow-acting, long-lasting effects. So that's sort of the canonical pathway where they induce changes that are impact the gene expression programs within neurons or within cells in the brain. And they can also act really quickly. So there's also receptors that act at the cell surface that bind to these, that these steroids bind to, that can exert quicker effects. Just to give you a sense of the broad scale, progesterone, both of these hormones, as I mentioned, are steroid hormones. They're originally synthesized from cholesterol, and they can be broken down. And metabolites of progesterone act directly on GABA receptors. So GABA is the major inhibitory neurotransmitter in the brain. GABA receptors are present throughout the entire brain. So when you think about sort of the scale of what can be impacted, it's really quite large because if you think about metabolites of progesterone acting on these GABA receptors, just about any system can be impacted. And then estrogen and progesterone receptors are present certainly in circuits that regulate maternal care. What we think of as the central site for maternal care is a region in the hypothalamus called the medial preoptic area. Certainly not the only region that regulates caregiving behavior. There's never one region, but the reason we call it the central region is because we think that it coordinates caregiving behavior through its projections to other brain regions, and it receives sensory inputs related to infants from just about every sensory modality. And so it interfaces with circuits that are involved in motivation, circuits that are involved in social behavior more broadly, and obviously circuits that will be involved in the direct behaviors that are related to caregiving. And so, how do these hormones impact the brain? Well, I think truly I think first I'll say, I think we're really just at the beginning of understanding the answer to that question.
Yeah.
But I think truly they impact every aspect of the brain. Within maternal neural circuits, they're going to increase responsiveness to infants. But even outside of those changes, we know that there are massive structural changes that are associated with pregnancy. So actually, for example, a relatively recent neuroimaging study came out of Emily Jacobs' lab. Really, really cool study doing dense neuroimaging throughout the entirety period of pregnancy and up to 2 years postpartum. And I think they found hundreds, I think it was something like 400 regions changing, and changes in gray matter. I mean, really, really large structural changes that could be seen, with the variability in change being substantially larger than what would happen throughout any other period of time in the lifespan of that same length. So these hormones are acting on receptors that are located throughout the brain, impacting
Right.
just about every system imaginable,
Yeah.
in addition to systems that regulate caregiving.
Yeah. So, we'll talk about maternal care behavior and some of the core circuitry involved in that. But the point you're making is there's basically a wholesale rewiring across more or less every region of the brain. It is a truly a massive change that happens.
Massive.
And so you've got these hormones, estradiol, progesterone, other ones, they get into the brain. These are fat soluble. So they get into the brain and they get inside of neurons. So they can both act at
Yes.
receptors on the cell surface to do what we might call fast changes, maybe akin to like what a neurotransmitter does to change neural activity right now, basically. But they can also go inside the cell, inside the nucleus of neurons in the brain, and then change transcription of genes, the turning on and off of genes. And that's really going to be the type of change that's slower, and it's resculpting the circuits literally. Like new synapses will get built or taken down. Things are being physically rewired. The behavior of the neurons, the activity of the neurons is being changed because these hormones get all the way inside of the cells to actually change what they do.
That's exactly right. And you can think about these changes we can think about the rewiring in terms of synaptic plasticity and structural changes, but you can also think about they can really change the way cells function. So
Mhm.
once you're impacting the expression of genes, and I should say, I meant to say this before, but when we talk about estrogen receptors and progesterone receptors inducing these long-term effects, there are many, 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 many genes that are responsive to these hormones. So it's not just genes that would be important for things you would think of as being specific to caregiving, but
Right,
I mean,
right.
we're talking about, basic signaling molecules that respond to these changes. And so you really can have just about every neurobiological and physiological system impacted.
Yeah, yeah, yeah. So changes throughout the brain, changes in the core circuitry that's directly related to maternal care, but changes, wholesale changes,
Mhm.
many circuits all over the place doing all sorts of different stuff. I would imagine one thing I want to say up front here too for people is to connect this to a lot of other things that we often talk about in the podcast. So estradiol and progesterone, can you talk about where they come from in relation to cholesterol?
Sure. Yes, they're synthesized directly from cholesterol in a biosynthesis pathway. And actually, you know, we so there are many, many different pathways. I won't go through all of them,
Mm-hmm.
but they can sometimes be they can be originally derived from cholesterol, but many of these pathways also interact with one another. And so there
Mm-hmm.
are many different pathways to produce estradiol and progesterone. This is a simple way to
Yeah.
say it.
So from cholesterol, you go to pregnenolone, then to progesterone, and then there's many different branching paths you can take, and some of those paths then hook into each other. But all of these hormones, or the ones we just mentioned, are ultimately derived from cholesterol. And
Mhm.
so these are all fat soluble things. They can get through the blood brain barrier, et cetera, et cetera. There's, it's obviously a huge change. It's complex. There's a lot to orchestrate. There's a lot that needs to go right. There's probably a lot that
Mhm.
could go better or worse under different conditions, and a lot of places where things can go wrong. What are some of the common ways that the pregnancy or the changes in the brain in particular of the mother might get disrupted even though she's still going to give birth and have the babies and stuff? So what I'm thinking of here is, are there common stressors that a pregnant female might encounter that are going to start to affect the rewiring of her brain and what the hormones are doing and all of that stuff?
Yes, absolutely. Stress is a major predictor of mood disorders throughout pregnancy and in the postpartum period. And I really think, again, we're sort of just in the beginning stages of understanding the complexities of how stress really does affect hormones, and also how hormones can affect our stress reactivity, because all of these things are sort of able to interface with one another. So we know that stress during pregnancy, as I mentioned, can produce long-term changes in terms of mood regulation. It can, during pregnancy and in the postpartum period, are one of the systems that's being altered by these hormones is the HPA axis, the hypothalamic pituitary adrenal axis, which regulates our stress response. And so experiencing chronic stress while that system is sort of being functionally altered can produce these long-term changes in stress reactivity, and, as I mentioned, in mood disorders as well.
And what would be like common stressors that a rodent mother like would encounter? How do you guys induce this in the lab? And how does this tend to play out?
So outside of the lab, I would say common stressors could be, difficult, low availability of food,
Mm-hmm, mm-hmm.
exposure to predators, and those sorts of things. In the laboratory, there are lots of different ways to model stress. Probably most of what we know about stress effects on pregnancy and the postpartum period in laboratory animals is actually modeled by just giving chronic injections of corticosterone, the stress hormone. And one of the reasons that people do that is that with many different types of stress exposures, you could get this elevated level of corticosterone. And so if you just give injections of corticosterone repeatedly over a period of time, you can sort of model that and see what effects will result from that. Um, there are, as I said, many different ways. Some models look at chronic restraint stress or physiological stress, so sort of restraining animals in ways that makes them uncomfortable. Mild unpredictable stress is also a really common model with changes in temperature or sort of tilting the cage. In my lab, we focused on social stress, which has been, I think, probably studied less in terms of the various models, but in terms of thinking about the clinical literature and what we know in humans about predictors of mood disorders in the peripartum period, psychosocial stress is a huge factor and one of the best predictors for the development of mood disorders. And so we were really interested in social stress. And so how do you socially stress? I mean, how do you socially stress mice, I guess could be a question, but how do you socially stress pregnant or postpartum mice is sort
Mm-hmm.
of a whole nother question. So generally speaking, many of the stress models that are used in laboratory animals are conducted in male rodents, and it's very easy to stress male rodents socially because other male rodents are stressful to male rodents. So
Yeah, they're
um,
territorial, so you can just expose
exactly.
them. Okay.
Exactly. And they fight. So actually I would argue most of what we have learned about the neurobiology of depression in terms of the role of stress in this altered circuitry is really from a paradigm called the resident intruder or social defeat paradigm, or the resident intruder stress
Gotcha.
paradigm.
You basically let two males fight each other. One of them wins, one of them
Duke it out.
loses. Yeah.
That's right. That's right. Exactly. And so this is it's a great model because it is a model of psychosocial stress. However, females don't fight with one another, and so it's not easy to translate into female rodents, and therefore, I think much of our understanding of how stress interacts with
Mm-hmm.
hormones,
Mm-hmm.
ovarian hormones, is really limited. So
Yeah.
there is one loophole to this model, which is that female rodents are generally not aggressive. The one exception is when they're lactating. So when females
Mm.
are lactating, rats and mice, that is when they display aggressive behavior, and they do so to defend their nest.
Right.
And there's great videos of this on the internet of just sort of wild rats that you can watch just fighting off snakes and things like that to defend their young. But the reality is that one of the biggest sources of potential threat for a postpartum female rodent is really a male conspecific.
Mm.
So as I mentioned earlier, most laboratory, or certainly most of the laboratory rodents that we study, but most mammals more generally, moms are the primary and the only caregiver. And so we call these uniparental species. And this is true for laboratory mice and rats. Moms do all the caregiving. Males are infanticidal, so they will actually kill pups if they encounter them. And the reason that they do this from an evolutionary standpoint is that when mom is lactating, she is not cycling. She's essentially in
Uh,
chronic diestrus. And
uh,
so if males want to mate, they encounter a female, she won't be receptive unless the pups are no
right.
longer
It's
around.
very much like other species, like lions, for example, do this type of
Exactly.
thing when the new male takes over the pride. I see. So while she basically has the pups, she won't be cycling. She won't be receptive to sexual advances from males. So they just kill the pups, or do they eat them? Do
Yes, they will kill and eat.
Okay. So
Yes.
the males are a huge natural source of stress. This actually I wanted to ask you about the natural sort of ecological behaviors here. So when a mother is pregnant and she's in her nest and then she has the pups, dad is not around at all. And then any males that would come are a threat and she's going to be aggressive towards them. Is that the normal rhythm here?
Yeah, that's exactly right. Actually, there's something very interesting here. So if you sort of, try to imagine a mouse world,
Mm-hmm.
you might be thinking to yourself, well, wait a minute, if males are always infanticidal if they're encountering pups, what happens if they accidentally encounter their own pups,
Right, right.
right? So what happens if they sort of wander away? Mating is promiscuous in rats and mice. And so, it's not as though they're sort of like living together prior to this. They just sort of encounter each other, and then, once females are pregnant, they kind of separate out onto their own. But so what happens is actually very cool. And this work has been done in a series of studies that were primarily conducted in the 80s and the 90s. But um, males will mate with females, and the sexual behavior actually triggers a time clock. And so now what's going to happen is about 21, as we get closer to 21 days later, around the time of birth, they are no longer infanticidal. And
Hmm.
my lab has actually looked across this time period. So if you test at 7 days, so 1 week, you present them with pups after sexual behavior, they're still infanticidal. If you wait 2 weeks, that's when you start to see a division where
Got it.
some males
Got
are
it.
are less so, but
It's pretty much tied
um,
to the time it takes for those pups to get
it's
born.
exactly.
So the
Yeah,
male will mate with the female. Obviously, the female gets pregnant, and then everything that comes from that will come from that. The male's now going to undergo his own types of changes that will modulate his behavior. But in this case, it's to eventually, at around the time that his pups will be born, even though he's not around and he's not going to experience any of that, he will become much less aggressive towards pups in general. And that's basically a way to help ensure that he doesn't kill his own pups.
that's exactly right. And I just want to say one quick um, caveat about all of this work, because you asked me about, do they eat, do they kill? They don't do any of those things in the laboratory. In
Hmm.
my experiments, for ethical reasons, we don't let the experiment go to that endpoint.
Yeah, yeah.
But we do know from semi-natural studies that they will cannibalize. That is the behavior. So in my laboratory, what we actually look for is the initiation of aggressive behavior,
Mhm,
and
mhm.
then we count that as pup-directed aggression.
Yeah. So one other thing that ties into this that's relevant to the idea of stressors on the mother during and after the pregnancy, but also ties into the differences in the male female behavior here is okay. So if there's no paternal care behavior, there's no daddy mouse who sticks around and does stuff, she's going to be in the nest. She's got to get her own food throughout the entire pregnancy, so the pregnant females have to really go out every day and get food and forage and do all that stuff.
That's right.
Okay, so it's not like
Yes.
it's not like parakeets where the male is going to bring food to the female in the nest.
No, that's exactly right. And actually, I would argue that in cases where you do see fathering behavior, that behavior has likely evolved because of the need for extra help.
Right.
So oftentimes when you see in mammals, when you see fathering behavior, it really just looks exactly like maternal behavior. Dads are just doing it alongside of moms. And that's not the case when you look outside of mammals. So oftentimes in other species where paternal behavior is a natural part of the species, you will see dads engaged in behaviors that moms don't do. They have their sort of own repertoire of behaviors. But in rodents, absolutely, you start to see these behaviors, and these behaviors that really mimic maternal care. And we can even get these behaviors to occur in the laboratory. So as I mentioned, in sort of the real mouse world, the real rat world, dad would never encounter mom again, probably, after she delivers. But in laboratory conditions, if you just keep the pair together, and you take mom out, dad will actually display pup retrieval. He will sort of crouch over the pups. He can't lactate, so he's not nursing, but he'll get into a similar sort of posture. So all of that can be induced.
And so when a mother gives birth and she's under particularly stressful conditions, I'll let you talk about which examples are the most illustrative, but I know one of the things that has been studied here that can disrupt or inhibit proper maternal care behavior is food scarcity. So the pups are born, she's got to eat because she's got to produce milk for the pups, but out in the wild, right? This is wild animals. Their food is scarce. She can't always get the food she needs. Sometimes food is so scarce that she's not going to be able to take care of those pups at all, or at least not all of them. When you're under a stress of that magnitude, like food scarcity, the mom has to go out in the world. She can't find food for whatever reason. How does that start to look in terms of how maternal care is disrupted?
Yeah, that's such a great question. I oftentimes have said about maternal care that it's incredibly robust, that moms are excellent mothers or they're not mothering. And so really what I mean by that is under situations of immense stress, if there isn't enough food, the response will often be infanticide. So oftentimes, and we see this, we sometimes see this in the laboratory. If, for example, there's, chronic construction noises or things that are oftentimes unforeseen, you will see many litters sort of disappear. So if mom doesn't feel as though she is going to have the resources that she needs in order to produce a viable litter, this sort of most adaptive thing to do in that situation is to not spend resources and energy rearing the young. Lactation is a huge energy expenditure.
Right.
It's a ton of work to care for infants. So actually infanticide does happen quite a bit, both naturally as well as in laboratory conditions if the stress is great enough. And so, oftentimes I get emails from other scientists saying, I all of a sudden, we're losing all of these litters. And I think we have, a model that could be interesting for, aberrant maternal care. And often I will say, if you're fully losing the litter, this is a case in which, I see this as being an adaptive response actually, and not a poor maternal care scenario for this particular species. So that's how I think about infanticide.
Yeah. And I think there's a couple important things here. One, as gruesome as it may be, if we think about this just in terms of calories and survival, if the mom doesn't have enough calories to make the milk for the litter, she might as well preserve herself and get some of those calories back from the pups to go on and do the next litter, especially because this is a promiscuous species and she will basically just be able to go out and start mating again if and when, she can survive and get enough food, et cetera. So from an evolutionary perspective, it does make sense in the grand scheme of things, the way this works in mice.
Yes, absolutely. And as soon as she stops lactating, she will be cycling again, right? And so she'll have a chance to do that. And actually, this is a kind of on the other end of the spectrum example, but we're talking about these really extreme situations of stress in which there's not enough resources. And so infanticide is occurring. But on the other end of the spectrum, there are natural variations in the ways that in which moms can care for infants. And the best example of this is Frances Champagne's work looking at variability in licking and grooming behavior in rats. And she really extensively studied the patterns of licking and grooming across the first week of life, and together with Michael Meany, they sort of identified high grooming moms and low grooming moms. And to be clear, we often will sort of talk about low grooming moms as being the bad moms, or the moms that are, we liken to maltreatment or reduced caregiving. But really, low grooming is still, these infants are still growing and surviving and doing quite fine. So another way to think about this high grooming and low grooming scenario is when you look at these rats as adults, you actually see different reproductive patterns. So the female offspring of high grooming rats become high grooming moms. So there's this, first of all, this really, really cool transmission of maternal style.
Right.
They are not as good at breeding. They're not as good at sexual behavior if you look at their
Hmm.
behavior. And the low grooming moms, they seem to have a different reproductive strategy. They
Hmm.
don't spend nearly as much time with their infants. They don't spend as much time grooming, but maybe produce more litters over
Really?
their lifespan
So
because they have heightened sexual behavior.
interesting, interesting. So this is, I didn't really anticipate going here, but this is interesting. And I think there's something more general happening here, which is if we bring in the concepts of R versus K selection, which we normally think about at the species level, these are also present and variable within species. So to unpack what I just said for people, two different sort of modes that we often see in nature for reproductive strategies are what you might call more of the salmon mode. Let me just have as many babies as possible, but not invest so much time and effort into caring for them. And then the opposite, which is what we think of as human mode, basically. Let's invest a lot of time and effort in a relatively small number of offspring. What you're basically saying here is that within one species, within mice, the mothers themselves can exhibit these different behavioral strategies. One will spend a lot of time grooming and tending to her pups, but she won't go out and have as much sexual activity to produce more litters. And then another type of female will basically have the opposite pattern.
Yes, that's the interpretation of that work. Absolutely. One interpretation of that work. Yes.
Mm-hmm. So different behavioral strategies tied to the way that individuals were cared for by their mother. You know, the pattern of licking, how much time she was there, which itself is going to be due to the hormonal soup of changes that happen tied to all the stressors in the environment and all of that stuff. So this stuff all connects and it can feed forward to the next generation and
Yes.
propagate in an experience dependent way. One thing I want to ask you about here is the effects of maternal age itself. Has anyone looked at how, on average, maternal care behavior varies in between, a mother's first litter and last litter, or if she has a litter right when she becomes sexually mature versus it's delayed and she starts later in life?
Yeah, that's a really great question. Um, hmm. I I can't think of any studies off the top of my head that have looked at the latter part of your question. So just sort of thinking about maternal care right after sexual maturity versus a total delay and the first litter
Mm-hmm.
being later.
Mm-hmm.
There are, of course, many studies that look at maternal care across litters, in which you're sort of looking at the combination of experience but age.
Right, right.
Yeah, so that's a really good question. I'm not sure.
Got it. So perhaps no one's done that type of experiment. But I want to talk a little bit more now about the pup side of this and the developmental effects of different patterns of maternal care behavior. So we started to get at this a little bit, but can you start to give us kind of a broad survey of what do the pups tend to look like during the time that they're pups, but also when they grow up and they mature, from litters where you have normal, adequate maternal care behavior versus maybe a more neglectful mother, not necessarily one who's, starving and has killed her pups, but one who's just licking less, not around as much, etc. What effect does that tend to have on the pups?
Yeah, great question. So this has been investigated in many different models. Some sort of separating mom from infants to produce this reduction in maternal care. Others, like the grooming work that I discussed, where you look at natural variations in grooming behavior, or even there are studies in which certain laboratory conditions can push mice towards low or higher grooming. When you look at the offspring, I think the first thing that I've already discussed a little bit that to me is the most notable is that you see the transmission of the experience. So when you look at social behavior, particularly maternal care, females that were raised in a low grooming environment will also spend less time grooming their own offspring, and vice versa. Raised in a high grooming environment will spend more time. That pattern is also similar when you look at extended periods of maternal separation, um, or early, even something called maternal separation early weaning, where you sort of remove mom and pups for hours each day. And then as you get closer to the time when mom and pups could naturally be separated, you separate them a little bit early. And in those cases, you do see reduced maternal care in female offspring when they become adults and they raise their own litters. So to me, there's something really interesting in that in early development, you're learning and growing. you're a pup in this example. You're learning and growing many things. But one of those things is actually how you will
Right.
care for your
Right.
own offspring when you're an adult. And then, in addition to that, there's also work to suggest that there's differences in social behavior. They can be quite dramatic depending on how dramatic the early separation or maltreatment paradigm was. There's behaviors that we observe in rodents that we sort of liken to the human experience of something like depression
Right.
or anhedonia, inability
Right.
to experience pleasure,
So
and those sorts of things.
if we were just really boil this down in simple terms, the quality and quantity of maternal care that a mouse pup gets will affect everything from what we can just call its personality, its behavioral tendencies later in life, to its sensitivity to problems, things like depression-like behavior, anxiety, and so forth.
Absolutely. Stress reactivity.
Yeah. Interesting. What within one litter for one mother, I'm imagining, I don't actually know the answer to this. It's kind of embarrassing. How many pups do they normally have several pups, but what's like the normal range here? I used to work on mice. What's the normal range? And what do we know about things like birth order or what we might call social rank? The pups that get the most milk versus the least milk. There's going to be kind of a dominance hierarchy that forms, and some pups will get more or less attention and nutrition and so forth. Do we know much about how that influences how the adult behavior turns out for those pups?
Yeah, that's a great question. And actually, there's not a simple answer to how many pups they have. So it really depends on strain. So C57 black 6 mice, which are the strain that I work with and probably one of the most commonly used strains, I would say they probably have on average about 6, anywhere from 3 to 9 pups, with 6 being kind of the average. Other strains like CD1 mice can have really big litters, 15 pups or so. So it just depends. It depends on the strain. The number of pups in the litter can absolutely impact. I think probably the biggest, most easy to observe difference is how big the pups are. So obviously, if there are fewer pups, they get
more milk per
more
pup.
milk.
Yep.
Exactly. And if there are more pups, then that's less so. And I think, in terms of their development socially, there can be differences depending on litter composition. So if there are, I think the biggest example is if there's only one pup in the litter, you can have really dramatic reductions in social behavior and sort of dysregulated behavior in adulthood. With only females in the litter, you can have sort of mild changes. Only males in the litter, sort of mild changes. I know what I was going to say. The thinking about the composition, you can actually sort of, this is not my area of expertise, but I think it's a really interesting point. You can actually go a step back. And I think one of the most interesting lines of research that has been done is looking at placement in utero. So looking at male and female pups next to each other, or male and male pups next to each other, or female and female pups next to each other. And there's really been like a whole line of research looking at outcomes related to just the placement in utero development.
Wow. So the technology is there that they can monitor the pups in utero and then follow them afterwards and know who is who.
Right. You can know whether, sort of, it was a female and 2 males were on either side, or, it was 2 males, et cetera. And there's been a whole line of work that's looked at that with development across the lifespan. I'm trying to think of other examples of related to the question of litter composition. Yeah, I think there's been really quite a bit of work looking at variation in composition and how that can kind of impact outcomes.
Yeah. So obviously, it's a little bit apples and oranges because, mice have litters of several. They don't have usually one baby at a time separated by some more extended interval like humans do. So, birth order is a little bit hard to translate here, but litter size, right? Litter size, the bigger the litter is, the less milk per pup, the more competition there's going to be for, who gets to suckle and all of these things. So bird's eye view, these things matter. This will affect future behavior in the adult versions of these pups based on how many litter mates they have, whether they were next to two boys or two girls, et cetera, et cetera.
Yes, exactly. And actually, there is another interesting thing, which is moms. So, we typically think about the litter as a unit, right?
Mm-hmm.
Because from a maternal behavior standpoint, when you're observing maternal behaviors, most of the things mom is doing to all of the pups in the
Right,
litter. And to try to get at questions about, differential care for different pups is a really logistically challenging thing to do because
right, right,
you are then sort of producing extraneous problems by taking one pup out of the litter and seeing how she
right,
responds to that or something. So
right.
it's hard to get at from a sort of naturalistic observational point standpoint, and I think logistically it's hard to test. That being said, there has been research looking in grooming behavior and specifically at sex differences in who's receiving that grooming. And so
Hmm.
moms do tend to groom males more than they groom females. And this
Hmm.
has an impact on male specific development. And when we talk about male specific development in rodents, we're usually talking about male reproductive behavior. And
Right.
so moms spend more time grooming the males than the females. And in this series of studies, what was really cool that they did is they went in and they were able to simulate mom's grooming with a paintbrush.
Hmm.
And so they were actually able to look at sort of the causality of this effect and say, like, okay, well, what if we take a female and we groom her in a male specific way? Do we end
Wow.
up seeing changes in adult behavior, of course, in response to exogenous hormones? But that's a very cool line of
Right.
research
Well, I mean,
because,
what do they
yeah.
see in that case? So they do a more, from the maternal perspective, you're applying a more male-like stimulus to the female pup. Does she then behaviorally look more like a male when she grows up?
If given exogenous hormones, you can look at her mounting behavior, and you can sort of manipulate hormones in adulthood to look at reproductive behavior separately from the organizational effects of those hormones. And so, um, yeah, so there have been some studies looking at that. So I think for me, the way that I interpret that data is that um, when mom is displaying differences in caregiving behavior, they are functionally meaningful.
Right.
They do seem to have an impact on development um, that is sort of relevant for that particular species.
Yeah. Yeah. And so, we talked a little bit earlier about all of the sensory modalities that are involved in the mother pup communication streams that are relevant here. She can obviously smell the pups, and smell and odor are very important for rodents. She can see them, she's picking them up, she's touching them, she's feeling them lactate, all of these things. And then they can do these ultrasonic vocalizations. I want to talk a little bit more about those. Like, how does so with what you just said, the mothers will tend to naturally, spontaneously lick the males more than the females. How do they know who's who?
The urine, so the pheromonal differences between the males and females.
Got
Yeah.
it. So they can just smell it, and they
Mhm.
can clearly tell.
Mhm.
Well, so
Yes.
there's these natural differences in the mother's behavior based on the sex of the pup, and those things just seem to be coupled. If you actually mess with them experimentally, you can make a female look a little bit more male-like in terms of her future behavior,
Yes. Yes. Her future sexual behavior, I should be clear. Yes.
future
Yeah.
sexual behavior.
Yeah.
Interesting. Um, so one of the big things that impacts pup development, obviously, is what they get from the mom, literally in the form of milk, the calories, the nutrients, and other things that are going to be in that milk. There's lots of stuff we could talk about here, from antibodies and immune system stuff to pathogen related stuff to pharmaceutical drugs that might be in the mom because an experimenter put them there or they're in the environment. And one of the papers that you guys recently did kind of gets in this direction. Before we get to that, is there anything in terms of background we should talk about in terms of lactation and milk that will set this up?
Um, I think I mean, we could talk about, um, I guess just sort of, uh, ov from a really general perspective. I think we don't have a lot of information about how, drugs that, mom is taking transfer through the milk and
Hmm.
how they might affect offspring. To me, this is a really huge sort of gap in our understanding. And, the standard ways that we tend to look at this is, looking at what mom's dose is and then collecting milk from mom. And this is in human data, collecting milk from mom and then asking whether or not the drug has transferred through the milk. There are all kinds of different things that can impact drug transfer, that have to do with, the lipophilicity of the drug and the pH. And I mean, there and individual variation in mom's production. I mean, there's many, many, many things that can go into this, but essentially we tend to look at mom's dose. We tend to look at the amount that we find in the milk. We look at the amount of um, milk that's consumed by the infant, and then we can calculate the relative infant dose. And that is, um, as far as I can tell, I should I should give the disclaimer that I don't do any research at all with humans, but as far as I can tell, that's sort of how we're determining the transfer through the milk, and then from there we're making decisions about whether that transfer seems to be a large dose, a small dose, etc. And there have surprisingly been not very many studies that have looked at these same questions in rodents, which does surprise me because it's obviously easier to go to the very next step in rodents, which is to then ask, okay, it's in the milk, is it in the developing brain? And if it is in the developing brain, are there any effects of exposure in the developing brain on the offspring, not only in the early life period, but of course, in rodents, their lifespan is much shorter. It's much easier to say, do we see changes in adulthood that could be causally linked back to exposure through the milk? So I think there's a lot of potential for asking those kinds of questions.
Hmm. And so, before diving into this paper, can you give us a sense here too for some of the timing? So, when a mother gives birth to a litter, how long is she managing that litter? And how long is this sort of peripartum, postpartum period where the pups are born until weaning, say?
So in the laboratory, it's really short. It's 21 days. We do that as experimenters. We call the process weaning, but really we are directly separating mom from
I
the
see.
infants. If we were not to do that, I think the pups would just try to nurse forever. And I'm sort of laughing when I say this because for a while, I did some experiments in a semi-natural environment where we had rats that were pregnant and postpartum living in these really large enclosures. And I can tell you in those cases that those pups never stopped. They were pinning mom down
Hmm, hmm.
to still
Yeah.
get milk from her when they were much, much, much older than 21 days. So
Yeah. Yeah.
so certainly I think there can be some probably natural variability in that. But in the laboratory, um, the laboratory standard is pretty much always between 21 and 25 days. And so during that period of time, from a developmental standpoint, to give you a sense, the pups are growing very, very rapidly, but they're still prepubescent. So puberty happens in mice from 35 to 45 days or so. So at day 21, they're still pre-puberty, but they're big enough to be able to eat solid foods on their own. And you know, they have their all of the other things that they don't have when they're very tiny in terms of their limited sensory and motor capabilities are no
Hmm,
longer a
hmm.
concern. And so at that point, they're mature enough to live in their own cage without mom.
Got it. So we can call that the mouse adolescence. Maybe in the wild, when they get out of the nest, they're pretty much ready to go in terms of adult and reproductive behavior. But there is this in between period, some amount of
Mhm.
time in between weaning and then doing, fully mature reproductive behavior out of the nest. Okay, but we're talking 21 days ish. That's what you guys do in the laboratory. Um, Set up this new paper for us. Why? So, just to give people the punchline here, they gave mothers psilocybin. Well, like, why did you even think to do this? And what was sort of the motivation for looking at this particular intervention in the postpartum period in rodents?
Yeah, so I think the reason why we did this, there's not one reason. There's so many reasons, but I think I was really excited for this project because as I mentioned earlier, I'm really interested in understanding experience-dependent plasticity in the postpartum period. And I focus primarily on how that plays out with respect to caregiving behavior. But more recently, I've been interested in whether or not peripartum physiology confers a sensitive period for experiences more broadly. So positive experiences, negative experiences, et cetera. And that's what really got me interested in sort of stress in the postpartum period. Um, and in that line of work, I've been very interested in trying to build a model of stress in the postpartum period that will have translation for our understanding of the neurobiology of postpartum depression, postpartum anxiety, et cetera. And so my mind was already very much in this. Let's try to understand how experiences, be it positive or negative, produce long lasting changes in behavior. When it was actually a student in a graduate seminar I was teaching brought in a paper about the possibility of using psilocybin for postpartum depression. And at that point, I had been aware that, psilocybin and other serotonergic psychedelics were looking really positive in terms of their potential for producing antidepressant effects. And I
Hmm.
always feel as though anytime we have something that is showing a lot of potential as a therapeutic, we should include the postpartum period, right? Particularly given that there's such a high risk, such a really true vulnerability for the development of mood disorders in this period to begin with. So this is sort of how the project got started. But I think what got me really excited about it is that um, psilocybin and other psychedelics have some unique characteristics as treatments that are really, really well suited for the postpartum period.
Hmm.
So, for example, time is of the essence, right? So we talked about the
Right. You want a
importance
rapid
of
acting thing.
bonding, rapid acting, exactly. Um, Chronic use is really not a problem, right? Because it's a sort of a fast acting effect often after one single dose. So the idea would be that, we wouldn't have to worry about drug transfer through the milk. You could take one dose and then wait for it to clear and resume breastfeeding
Got it.
and things like that. So I was, I thought we were very excited about this. And
Right. So I guess,
I got,
yeah.
yeah,
So maybe one way
sorry.
of saying this to make sure I understand is the postpartum period is a sensitive period for females. We know that in humans, postpartum depression and mood disorders are very common. We talked earlier about how this is like a developmental sensitive period
That's
for
right.
females. They're just these things happen. So you've got this window in which mood disorders become more common, and in contrast to something like an SSRI, say, where there's going to be a delayed effect. You usually need to wait 1, 2, 3 months to start seeing some effects if they show up. You need to take it chronically. Psilocybin was interesting to you guys because it seems to have rapid acting antidepressant effects even after one dose in some of the studies that have been done. And so maybe you thought you could solve sort of the mood problem for the mother, but do it in a way where you're using a low dose of something that doesn't require chronic intake, and therefore you're going to minimize exposure to the pups in the experimental setting.
That's exactly right. Yes.
Okay. Okay. So set the experiments up for us. You gave
Yeah.
psilocybin only after the pups were born.
Correct. So as I was mentioning earlier, I was interested in stress in the postpartum period. And so our model of stress in the postpartum period is very similar to the social defeat paradigm, the resident intruder stress, where we expose moms to a male conspecific, and she defends her nest, which she's very good at doing. But we had previously found that if you kind of repeat this process over many days, it becomes a repeated stressor for mom, and it begins to dysregulate caregiving behavior. So our thought was, let's give psilocybin in the postpartum period after this stressor to see if it can rescue the effects of repeated stress. And this is very similar to many other studies that have been done with psilocybin and other psychedelics outside of the postpartum period, just looking at, can the effects of stress be rescued?
Okay, got it, got it. So that all makes sense to me. So What is like the first experiment you did here? And what did you basically start to see when you gave psilocybin in this context?
So our first experiment was very basic. We used a 2x2 design. So we really wanted to see not only would psilocybin rescue the effects of stress, but also were there any effects of psilocybin in and of itself. And so
Hmm.
we had postpartum animals that experienced the repeated social stress, postpartum animals that experienced no stress, just normal postpartum conditions, and then within each of those groups, we gave psilocybin or we gave saline, which was the vehicle control. And we did this on postpartum day 7, which was the last day of our stress. So I guess I should have said this before, but previously when we found these effects of stress, not only did we find dysregulation of caregiving behavior following stress, but we found that the dysregulation was sustained for at least 24 hours after the last stressor.
Hmm.
And so if you do this add the social threat every single day, you see the behavior following the social threat becomes dysregulated on postpartum. So you do this on postpartum days 5, 6, and 7. And then 24 hours later, you don't expose them to a male intruder. You just do a brief maternal separation, and
Hmm.
then you see how they respond to their infants. And we were finding similar effects. So we basically put the psilocybin in between those two. So
Right,
we
right,
said we give it on day 7 after their last experience with stress. 24 hours later, the next day, we were expecting to see
right.
um, caregiving behavior
Got
reinstated.
it. So to tie some things together, the presence of a male is inherently stressful to a new mother because dad doesn't stick around. Males are dangerous. They will kill the
Right.
pups under ordinary circumstances. And remind me, so how do you expose them to the male? Are you literally dropping a male in the cage? How does that look?
Yes. So so actually I'll set the cage scenario up too because our caging is a little bit unique for this paradigm. What we really wanted to understand was, how would mom respond to social stress in general? And we wanted to get at not only sort of what we would think of as the regularly studied disruptions in care, but we really wanted to see whether or not mom would withdraw from her infants. Social withdrawal is a major feature of depressive disorders, but also in the social defeat paradigm, many people have examined male social withdrawal after the repeated stressors. So we gave these animals essentially 2 cages attached with a PVC pipe in the middle. So mom has like a mansion scenario, if you will. She has one cage where she has her nest built, and that's where she spends a lot of her time.
Yep.
But she has another cage that she can basically hang out in with food and water, but with no infants.
Right. Got it.
And so what we did in the stressor is we enclose mom in just the nest cage where her nest is located by just putting a cap on the PVC pipe. And then we take the pups out because we don't want anyone to actually get injured.
Hmm.
And you don't really need the pups to be in the cage for maternal defense to
Got it.
occur. So we take them out to protect them. We put the male in. We just drop the male in for 7 minutes is the standard
Hmm.
paradigm.
She
And
sees
during
them,
that,
she smells them, and okay, they interact.
she chases after him.
Got it.
So
Okay.
B6 mice are not particularly aggressive. So we don't see, so with other strains of mice, mom will beat up the male. I mean, she will
Yeah.
kind of not,
Yeah.
there won't be any injuries by any
Yeah.
means, but
But
she
it's
will
it's
sort of
crystal clear.
pounce.
She doesn't want them in there. You can see
She
it.
does not want him in there. B6 mice don't show that level of, boxing, but they do sort of chase the male around the cage. And so the males are quite stressed from this as well. And so we do this for 7 minutes, and then we take the male out. We give her a couple of minutes to calm down. We open the portal so she can explore her full cage. And then 3 minutes after that, we put the pups back into the cage. At which point we call this the reunion. And then we observe their behavior for an hour. So we do a really extended observation so that we can see, how does she respond to her infants after being exposed to this stressor?
Mhm. And so, what are the behavioral manifestations of stress in the mother after the male's out of the picture? What is she doing differently that she wasn't doing before?
I love the way you phrased that, because going into this, what I was sort of expecting was that I would just see less maternal behaviors, but that's not what we see entirely. We do see her doing different types of things. So when we put the pups back into the cage, she does display pup retrieval behavior. It's not always perfect, or perfectly, coordinated, but she will bring the pups back to the nest. After that point, she will often display really sort of like rapid movements between the two cages. She will oftentimes dig, which is a stress behavior that we see. Sometimes she will become sort of inactive and grooming outside of the nest, which is very rare. Moms are usually always in the nest.
Hmm.
Sometimes she doesn't seem to recognize the pups as her infant. So I think in the paper we talked about this as treating pups like inanimate objects, and
Hmm.
that's what it looks like. There's a supplementary video that you can watch, but essentially she's so frantically digging. At first, you almost think she's trying to build a nest, but you can see she's just
There's
sort of
kind of no
rustling,
rhyme or
bedding.
reason to it. Yeah.
Exactly. It doesn't seem to be organized in any way. And if she is digging and she encounters a pup, the pup will sort of get
Swept in.
tossed
Yeah. Yeah.
aside. And so she's really focused on this task, which is very frenetic. And
Right. She's getting
we
distracted by tasks that aren't particularly relevant.
exactly,
Yeah.
exactly.
Hmm.
And will sit next to the pups. So I was sort of referring to this earlier, where usually once the mom is in the nest, she's going to start nursing because the pups are going to instigate that posture. But these moms will often sit and they'll keep themselves kind of to the side so that the pups really can't crawl underneath
Hmm.
them.
Hmm.
So she's really preventing them from nursing as well. So all
Interesting.
of those behaviors we classify as stress related. They're very
Yeah.
different than what you would see in a normal maternal care scenario.
Yeah. Yeah. So it's not subtle. The behavior is quite different. Yeah. It's not that she's completely usually ignoring the pups and not doing any maternal care behavior, but you see all this extra stuff and all this disorganization in her behavior.
Disorganized, I would say, it's if anything is the opposite. I don't think she's really she does spend quite a bit of time in the cage without the pups, and we do talk about this as being an avoidance or withdrawal. But I would say a lot of the behaviors that we see the moms engaged in, it's almost like they're trying to figure out what safety would be.
Right.
It would be my anthropomorphic interpretation, having watched many of these
Right.
animals. and
Right.
and so they're busy, they're very busy, there's a lot of
Yeah.
activity.
What do I do? Where do I go? I'm digging, I'm searching for something. I don't know what. That's what it looks
Hmm.
like. It's organized,
Yes,
it's frantic.
yes.
Yeah. Okay, interesting. So clear effect of the males. It's not a subtle effect at the behavioral level. Presumably, there's all sorts of hormonal changes and things like this. 7 days of exposure to the male. So it's 7 days of stress.
So,
Normally,
three
go ahead.
days sorry
Oh,
three
three
days
days. Okay.
Mhm.
3 days of stress in a row. And then typically the mothers look stressed out for an extra day after. And that's when you're going to give the psilocybin treatment or the control vehicle treatment in this paradigm.
We give it just before that extra day.
Got
That's
it.
right.
Got it. So the question is like, are you going to see that extra day of stress? Is it going to go away? Is it going to be different? That's what you're
Yes,
looking for. And
exactly.
so you give psilocybin. Give us a sense for the dose here. Is this a big dose, a medium dose? How would you describe it? And then what do you see?
Great question. We use 2 milligrams per kilogram, which is a standard dose that's been used in many studies with psilocybin in mice, in males and females. Actually, we chose this dose because it is a dose that so many other laboratories have used. And what we found is, well, the first thing that we found in that 24-hour period is that nothing really changed.
Hmm.
So we didn't see any differences
Mhm.
in the stress related behaviors for the moms that experience stress if they were treated with psilocybin.
Got it. The big hope would have been it all goes away.
Yes. So we had two hopes. Our first hope was that hope that 24 hours later we would see differences. And our second hope was that things would be different at an extended time point. And so just to sort of
Right. Rapid
give some
acting and
background.
long lasting effects. That's what you were,
Exactly.
yeah, yeah.
Rapid acting and long lasting. And when we designed this, the way that we designed this, again, I sort of think about some of these postpartum loopholes as I thought, well, this will be really perfect because we also knew right from the get-go that we wanted to look at offspring. And so we said, okay, what we'll do is, the testing involves pretty brief mother-infant separation. So we weren't concerned about that in terms of long-term effects on offspring. We'll look 24 hours later, then we'll leave them completely undisturbed. But we know from the literature that not only should we get these fast-acting effects, but we should get these long-lasting effects. So after the pups are weaned at day 21, it gives us the perfect chance to finally go in and do standardized behavioral testing for things that aren't related to maternal behavior. So we
Right.
could look at, how do they respond in a novel environment? They would look at their stress reactivity. We could look at behaviors that are indicative of what we would call behavioral despair or depressive-like behavior. And we would do a standard test battery at the 2 week time point. So it'd be roughly 2 weeks after they got their injection. And so our first hope was we'll see reinstatement of caregiving behaviors and everything will look normal at the 24-hour point. And then our second hope was 2 weeks later, we'll see antidepressant, anti-anxiety effects of the treatment.
Got it. So one day later, you don't see what you hoped for. Her behavior
Mhm.
is still looking frantic, let's say. It's not looking that much better or not better at all. I'll let you tell me. And so I guess that's observation number one. The psilocybin didn't
Mhm.
seem to help in the short term in that first 24 hours.
Correct. No differences between the psilocybin and the controls.
Got it. And then you wait. And then what do you see?
And then we wait 2 weeks later, and then we run the animals through a test battery. So again, just to set this up, we did tests that are related to cognition. We did tests that are related to anxiety-like behaviors. We did tests that are related to depressive-like behaviors. And what we found is across all of these tests, we did not have any antidepressant or anti-anxiety
Mhm.
effects. But we did have, sorry, you're going to
I was
say.
gonna, so, but correct me if I'm wrong.
Mhm.
In other conditions, you give a mouse psilocybin, you do see antidepressant effects in these types of behavioral paradigms?
Yes,
Yes.
yes, yes.
Okay.
Sorry, yes,
So it's not, you're
yes.
not seeing it in this context, but other experimenters do see this antidepressant like effect, which is akin to what we see in humans, we think, in these types of behavioral paradigms. So I think that's important.
It's yeah, absolutely. Yes. Thanks for should have said that. And this is why we chose these particular behavioral tests. We were really looking for what has been done, what has been found, and let's create a small test battery to get at some of those things. And that's where we had our most surprising outcome, which was that, we didn't see, we did actually see significant changes associated with psilocybin, but they were in the inverse direction that we expected. And we saw them regardless of whether or not animals experienced stress.
Wow.
So across all females that were treated with psilocybin, the biggest effect that we saw was an anxiogenic effect
Hmm.
or an increase in anxiety behavior on two tests that measure exploration of a novel
Mhm.
environment. Novel environments are
Yeah.
really stressful for mice.
So if I'm hearing you correctly, there are mothers you expose to the stress of a male being present that disrupts their normal behavior, that makes them more anxiety like in their behavior in general. You give them psilocybin, it does not help with that, and it actually makes it worse to some extent. And you also see the same type of what we can just call an anxiety like response to mothers who are not even exposed to that stress.
That's exactly right. And we actually didn't, to be clear about this, we did, I mentioned we had a 2x2 design. So when you have a 2x2 design, essentially you are looking at two independent variables and you can ask whether or not each one of those variables impacts your outcome, but also whether there's an interaction between
Yeah.
those variables. So we were looking at stress in the postpartum period, psilocybin exposure, and then, of course, the question of whether the two interact, which was our hypothesis. We would have hypothesized that they would interact in such a way that we would see animals that got stressed, but psilocybin behave differently. And what we saw instead was main effects of each of we saw a main effect of psilocybin, and we actually did not see a main effect of stress. And really, throughout the paper, we never saw an interaction between stress and psilocybin. And so, yes, I do think the animals that were stressed are worse, but I think what we were even more surprised to find is that even in the absence of the male stress exposure, we
Yeah.
still saw changes in the anxiety-like responses of the mice
Yeah.
that were exposed.
Yeah. And how do you start to like interpret this at an ecological behavioral level? Like I would start the way I start to think about this in my mind is, this is a sensitive period for the mother. She's naturally going to be more prone to anxiety because it's part of the defensive aggression, detect threats in the environment state she has to be in as a mother. This is basically what her brain has just been rewired to do. And just as the male is a novel or unpredictable stressor, it's an external thing coming in, disrupting, the coziness and the consistency of the nest environment. That's stressing her out and disrupting her behavior. The psilocybin itself, you could imagine it being a similar thing. She's not expecting this. All of a sudden, the quality of her experience is changing. She could be perceiving that as an external threat, almost like the male.
Absolutely. And actually, we do know that even though I mentioned before that psilocybin is known to produce antidepressant anxiolytic effects in rodents, we also know that it produces an acute anxiogenic effect. And so, meaning that within the first few hours that an animal is exposed to psilocybin, there is sort of a very very short rapid increase
Right.
in their anxiety responses.
Yeah.
What's different about our model is that during that experience, infants would also be there. So I think
Hmm.
the even just the context of how the psilocybin is administered would be
Right.
really different. And then,
Right.
of course, add to that all of the changes that are happening in the
Right.
postpartum
Right.
period, and this completely, I mean, the plasticity is, um, as we talked about before, incredibly robust
Yeah.
throughout pregnancy and in the postpartum
Yeah.
period. And now you're giving a drug that
Right.
mechanistically dramatically alters
Right,
plasticity.
right, right. And it's
Um,
hard not to think about human parallels here. Anyone with experience with psilocybin who's looked into this closely will know that even when it has antidepressant effects, even when the trip is good, very often for a few minutes when you're coming up, so to speak, there's this, oh God, like anxiety moment that often goes away, but not always. And when it doesn't go away, that's typically because something is instigating that. It's not difficult to imagine someone coming up on psilocybin and then thinking, oh God, I'm tripping on mushrooms, but I have to take care of my kids. Like when you think about
right.
this response, you're supposed to do these things in a safe environment where you don't have to think about responsibilities and threats and that stuff. So it kind of all is making a lot of sense to me from a human perspective as well.
Yes, absolutely. And in the first experiment of the paper, we gave those injections and we looked at the behavior 24 hours later. So we really didn't see the interaction between mom and infants fall immediately following the psilocybin exposure. But in a later experiment in the paper, we did, and you absolutely do see increases in stress behaviors. Um, Regardless of, in the second experiment, there was no stressor. It was just exposure to psilocybin. And you can see that mom is in a displaying or displaying stress related behaviors, excuse me, and not displaying caregiving behaviors. And I absolutely think about it like those pups are probably crying. They're probably
Right,
emitting
right.
ultrasonic vocalizations, and from a sensory standpoint, how must that what is that experience
Right.
like?
Imagine tripping on mushrooms while your baby's crying in the next room. Yeah,
Doesn't
it's
sound
not going
good.
to be
Yeah.
good.
No.
Okay, so there's two ways in principle here I can imagine the psilocybin affecting the pups. One is through the mother's behavior, which is altered. So obviously, if this is altering her behavior, it's going to affect how she interacts with the pups. That's going to affect the pups developmentally. The
Yes.
other potential avenue here is that some of it might get into the pups from the milk. So let's talk about the pups now. I'll let you guide us in terms of where to go next, but are they getting exposed to it in the milk at all? And, how does this transition into how they are affected later on in life?
Absolutely. So we did not separate mom and infant after the injection, as I just mentioned, and so we assumed that there could potentially be transfer. We did conduct an experiment to test that assumption, and we looked at. We actually didn't look at the milk. We looked directly at the brain of the infants to see if we could find psilocin, which is the active metabolite of psilocybin that really is producing all of the effects. And we did find psilocin in the brain of pups, so it does seem as though it's able to transfer through the milk and reach the brain of the developing offspring. This would have been on postnatal day 7. So mom received one exposure, so pups would have received one exposure. The amount of psilocin that we see in the brain is much less in the pup than what we would see in the mom, which again is what we would expect. And then we followed pups in adulthood to see whether or not that exposure that happened at postnatal day 7 produced any changes in their behavior. And to look at their behavior, we conducted a behavioral test battery very similar to what we did in mom, looking at, again, cognitive behavior, social behavior, anxiety like behavior, depressive like behavior. And what we found in the pups, we looked at what we found in the adult offspring, because they're adults at this point, and we looked at males and we looked at females, is we found overall an increased risk for what we would call behavioral impairments. So when we do these batteries, we do look at the individual results of each test, but to get a better idea of behavioral change across the test battery, we also use and we z-score each individual behavior, and then we look at the sort of average of their behavior across all of those tests, and we see increased behavioral impairments associated with that single exposure in males and in females. And of course, this is, just to be clear, this is more than 100 days after that initial
Right.
single exposure.
Right. So this is a lasting effect. This is, they're developing differently as a consequence of
Mhm.
having been brought up in this fashion related to the psilocybin treatment of the mother, which is changing her behavior. So like
Mhm.
their whole childhood effectively has now been different in terms of how they interact with
Altered.
their mother.
Mhm.
Um, so this is a pretty big effect. What do you think sort of the headline is here in terms of how you think about and interpret the results for the mice themselves, but also in terms of the implications for human beings?
Just one quick thing I want to say before, that's a great question, but you brought up that, mom's behavior is affected by the psilocybin treatment, and pups would be exposed directly to the psilocin, and all of that is absolutely true. And one of the things that's really difficult is to disentangle whether or not the effects you see in adulthood are related to the exposure to the
Right.
psilocin or to mom's behavior. I mean,
Yeah.
this is a constant question in my field. And there are many ways of addressing that question. But what we did another experiment in this study where we exclusively exposed pups to psilocin. So we did
Oh.
not expose mom at all. And we actually did this within litter. So within every litter,
Oh wow.
some of the pups were injected with saline and some of the pups were injected with psilocin. We did this on postnatal day 7. So the same time point. And we did observe maternal behavior just to make sure the injections didn't throw mom off, but they did not. We left them totally undisturbed. And once again, 100 days later, we looked at their behavior in the same test battery and we replicated the effects. Not 100%. So there clearly is some probably influence of mom. But
Right.
I think what really stood out to me that's quite robust is in both experiments,
Yeah.
we found a significant uh, change in sucrose anhedonia.
Got it.
Just in one specific test, we saw
Right.
that the same result both times, which is
Yeah.
anhedonia is a
Right.
hallmark of depressive behavior.
Right.
So
Anhedonia, lack of pressure. They're not liking the sweetness as much. We think that is a
exactly.
depression-like thing. Got it. Wow. So if you would have told me that the psilocin completely recapitulated the results, I would have been skeptical because mom's behavior has to have an effect. We know it does.
Right.
But
Yeah.
what you're saying basically is giving psilocybin, which turns into psilocin, directly to the pups alters their development almost as much and in many of the ways as giving it to the mom whose behavior is then disrupted and then has the effects on the pups.
That's exactly right. And we were surprised, but I am certainly less surprised now having read a handful of papers that came out relatively recently. And there are some older papers that suggest similarly that serotonergic systems are developing during that window
Right.
between
Huh.
postnatal day 4 and postnatal day 8. And so other work has shown that administration of SSRIs at that time or manipulations to the serotonergic system do produce these long-lasting changes
Got it.
into adulthood. So there is precedent for that idea, but it was very surprising to see such a similarity in behavioral change in our psilocin only
Yeah.
experiment
Yeah.
compared to our mom experiment.
Yeah. It's an
Yeah.
intriguing finding because I actually overlooked that experiment prior to talking to you right now. So you just told me for the first time. I would have predicted that most of the effects were coming by way of the mother, in part because the mother's behavior we already know is important, but also because the dose is going to be so much smaller for the pups. But apparently, even though there's going to be a drop off in the dose, mother to pup through the milk, it's enough. And based on what you just said, it actually does make sense. We know that the serotonin system is developing a lot at that time,
Mm.
and that other manipulations of serotonin with SSRIs or other things have that type of effect. So it all did seem to fit in the end when you considered it in the context of all that other literature.
Yes, absolutely. And I think, in terms of thinking about the bigger picture of some of this, which is the other question they asked, I think, you know, one of the things to think about in terms of the maternal period is that all serotonergic systems are also very much a part of the reorganization that's happening in the maternal brain.
Mm-hmm.
And so, you know, in some ways, I think one of the big messages of this study is thinking about how administration of these plasticity inducing drugs that act on serotonin systems can have different types of effects when they're administered in developmental windows in which serotonin itself is being reorganized or signaling is being affected. And I think part of that piece is that we know stress can largely affect serotonergic systems as well. And so I think there's a really complex interaction between stress, between serotonin. I can't remember if I mentioned this earlier, but ovarian hormones like estradiol and progesterone directly regulate serotonin signaling as
Mm-hmm.
well.
Mm-hmm.
And so all of these things are changing in both the postpartum period and in the early postnatal period in this model. And that can not only lead to long-term effects of the psychedelic, but also really change the way, in this case, psilocybin produced its effects. And so I think there are a lot more questions that we need to answer about exactly how this is happening. But I think it also sheds some light on really how much we don't know about the interaction of some of these complex systems during these developmental transitions.
Are you guys doing any more work coming up connected to this study? Or is there any follow up to this one?
Yes, absolutely. There's so many things that I want to do, and there's so many things that we've started to do. But I think, first and foremost, I think we have a lot of questions about what's happening in the brain. So this paper was primarily focused on behavior. It was incredibly labor intensive and extensive series of behavioral experiments, but we didn't look in the postpartum brain or in the brains of offspring in this study. So I think we want to have a better understanding of how psilocybin is affecting the maternal brain, and whether it's, in ways that we might expect given psilocybin's effect in non-pregnant animals, or whether it's in really dramatically different ways, and ultimately try to link that back to the findings that we have with behavior. And then
Yeah.
I think, sorry, okay.
Oh, no, no. I'm just wondering how can you give us a sense for how big the effect was? So like the anhedonia in the pups, were they like 10% less sensitive to sucrose rewards, half as much? Like how big was this effect?
Oh, that's a good question. Well, I believe the effect size itself was pretty large. I'm not sure percentage
Yeah.
wise.
But it wasn't like super
Um,
subtle. It was a pretty clear effect.
it was a well, I mean, yes, it was absolutely it was a clear effect with a large effect size, and a low p-value, and replicated across, you know, two completely different experiments
Hmm, hmm.
essentially that were conducted. Actually, were conducted in two totally different laboratories, because
Mm.
in the middle of the study, my lab moved to a
Okay.
different location. So, it was completely different people handling the animals,
Yeah. Yeah.
completely,
And
everything you can imagine changed, and it replicated. So I think it's robust in that sense that we've seen it twice, and that the effect
yeah.
size was
And
large.
I hope this isn't a non sequitur, but I do know that like even the sex of the experimenter can affect how stressed out mice get. Was there any t- was it males or female humans doing the experiments?
Uh, all
Both,
sexes
both. Okay. Yeah.
involved
Yeah.
in the experiments, across all of the experiments that we've run. Yeah, and lots. My lab is. I have a ton of really fantastic undergraduate students, many of which are authors on the paper, that really spent a lot of time helping to run these experiments. And so there were many different people that were involved in the collection of the data.
Mhm. And what's your big takeaway here? What is the message in terms of humans and human mothers, and whether or not, obviously there's implications here for like, should a woman consider psilocybin for postpartum depression? All of those things. What would you say to people in terms of the human implications here?
I would say, well, first and foremost, I would say that mice are not humans, of course, but I do think that what we have uncovered here is, I think it's pretty clear that psilocybin can produce different effects in the postpartum period, possibly related to postpartum hormonal milieu. It can interact potentially with serotonergic changes that are in the brain. And so I think that this doesn't mean that psilocybin won't, should be completely not considered in the postpartum period, or couldn't ever be a treatment. But I do think that it calls for some caution, and I think it calls for a lot more experiments to better understand how and why we saw these adverse
Yeah.
effects. We did also replicate the adverse effects twice. So I do feel like this was a really robust change that we see in these animals. But at the same time, we do also know that there is a, a a pharmaceutical company that is running clinical trials right now for a drug that it's not exactly the same thing as psilocybin, but it is relatively
Right.
similar to
Yeah.
psilocybin. And they're seeing efficacy in their early
Yeah.
reports with postpartum depression. And so
Yeah.
I don't, you know, there can be many reasons why we see differences
Yeah. What
in humans and our work, but
Yeah, I think I think what this really reinforces is that the effects of psilocybin are highly context dependent, and if we analogize your experimental setting that you had for your mice to humans, right? What the experiments you did would be more akin to a mother tripping on shrooms at home while her baby's
right.
crying in the next room, not going into a clinic where she knows dad's at home taking care of the kids, everything's okay, and now I can go have this therapeutic type experience. So we need to keep those things in mind when we do experiments in
Absolutely.
humans that show that psilocybin has efficacy, right? We take great care to make sure they're comfortable, they're safe, they've got nothing to worry about. That wasn't the situation that you modeled in mice. It was something that you would easily imagine was a very stressful experience for the mother.
That's absolutely right. And I think that's a really important consideration. And I think that, I mean, it's very possible that we could have seen different effects had we separated infants. So I think from the perspective of really trying to understand what psilocybin is doing in the brain and how it's altering sensory inputs, how it's affecting the HPA axis and how it might be interacting with hormonal changes. There's so many interesting questions that we can ask there. And we have the ability to manipulate these different things to see how it then produces effects on not only the behavior short term, long term, but also on the neural systems that are responding to infant cues.
Is there anything you want to reiterate from our discussion today, or any final thoughts you want to leave people with beyond what we just covered?
Not that I can think of. I think we covered many things. So, yeah.
I think this was excellent. This is a really interesting study that people were talking about. So I definitely wanted to get you on and talk about it. Thank you for agreeing to do it on fairly short order right before the holidays. This is the last one before Christmas. But if that's it, do you have a lab website or anything that you can point people to where they might go to learn more?
Sure. I have a website linked to my UC Davis profile. So if you search for my name and UC Davis, you can easily find my website where we have updates on things that we're working on, etc. And it was my pleasure to be here. Thank you for inviting me.
All right. Well, Danielle Stolzenberg, thank you again for your time.
Thanks.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.